Hello students, this is your instructor Abid Jan. I will proceed with the remaining part of sequence similarity searches from the course of bioinformatics. FASTA sequence format. The FASTA sequence format includes three parts. A comment line identified by an underscore character followed by the name and origin of the sequence. The sequence in standard single letter symbols. An optional asterisk symbol which indicates end of a sequence, and that may or may not be present. However, the presence of asterisk symbol may be essential for reading the sequence correctly by some sequence analysis programs. The FASTA format provides a very convenient way to copy just the sequence part from one window to another because there are non-sequence characters within the sequence. Pairwise sequence alignment. Sequence comparison lies at the heart of bioinformatics analysis. It is an important first step towards structural and functional analysis of newly determined sequences. As new biological sequences are being generated at exponential rates, sequence comparison is becoming increasingly important to draw functional and evolutionary inference of a newly protein with proteins already existing in the database. The most fundamental process in this type of comparison is sequence alignment. So this is a process by which sequences are compared by searching for common character patterns and establishing residue to residue correspondence among related sequences. Pairwise sequence alignment is the process of aligning two sequences and is the basis of database similarity searching and multiple sequence alignment. Evolutionary basis. DNA and proteins are products of evolution. The building blocks, nucleotides and amino acids of these biological macromolecules determine the primary structure of these molecules. These molecules, nucleic acid and proteins, can be considered as molecular fossils that encode the history of millions of years of evolution. During this time period, the molecular sequences undergo random changes, some of which are selected during the process of evolution. As the selected sequences gradually accumulate mutations and diverge over time, traces of evolution may still remain in certain portion of the sequences to allow identification of common ancestry. The presence of evolutionary traces is because some of the residues that perform key functional and structural roles tend to be preserved by natural selection. Other residues that may be less crucial for structure and function tend to mutate more frequently. For example, actioside residues of an enzyme family tend to be conserved because they are responsible for catalytic functions. Therefore, by comparing sequences through alignment, patterns of conservation and variation can be identified. Inferences that can be drawn from sequence alignment. Identifying the evolutionary relationships between sequences helps to characterize the function of unknown sequences. When a sequence alignment reveals significant similarity among a group of sequences, they can be considered as belonging to the same family. If one member within the family has a known structure and function, then that information can be transferred to those that have not yet been experimentally characterized. Therefore, sequence alignment can be used as basis for prediction of structure and function of uncharacterized sequences. If two sequences share significant similarity, it is extremely unlikely that the extensio similarity between the two sequences has been acquired randomly, meaning that the two sequences must have derived from a common evolutionary origin. When a sequence alignment is generated correctly, it reflects the evolutionary relationship of the two sequences. Regions that are aligned but not identical represent substitutions. Regions where residues from one sequence correspond to nothing in the other represent insertions or deletions that have taken place on one of the sequences during evolution. It is also possible that two sequences have derived from a common ancestor, but may have diverged to such an extent that the common ancestral relationships are not recognizable at the sequence level. In that case, the distant evolutionary relationships have to be detected using other methods. The three zones of protein sequence alignment. When two protein or gene sequences are very similar, biologists call them homologous. If your sequences are more than 100 amino acids long, the rule says you can label proteins as homologous if 25% of the amino acids are identical. For DNA, you will require at least 70% identity to draw the same conclusion. In simple words, 25 amino acids out of 100 amino acids and 70 nucleotides out of 100 nucleotides. Two protein sequences can be regarded as homologous if the percentage sequence identity falls in the safe zone. This is the safe zone. And the value of percent identity must be above 25. Sequence identity values below the zone boundary but above 20% are considered to be in the twilight zone. About 20% and less than 25%, where homologous relationships are less certain. The region below 20%, this is the midnight zone, where homologous relationships cannot be reliably determined. This is the bibliography. Keep calm and stay curious. All the best.